The ocean floor is dark, not the darkness of a room with the lights off, or the darkness behind closed eyes. This is the kind of darkness that has weight, cold, pressurized, absolute, and somewhere in that darkness, resting motionless against a patch of coral rubble, is an animal that should not exist the way it does. It sits still, completely still. And yet it is not stone, not debris, not shadow. In less than a second, without apparent effort, it becomes the precise texture, color, and pattern of the surface beneath it. Not approximately, not roughly, but with a precision that defies the biology behind it. Because this animal is colorblind, its blood is blue. It has 3 hearts. 2/3 of its brain lives outside its skull. It reproduces once in its lifetime and then, by design, it dies. Today, we're diving into why nothing about the octopus is normal. Not its body, not its brain, not the way it sees, adapts, sleeps, or ends its life. And the deeper science goes, the more unsettling the answers become. To understand what makes the octopus genuinely unlike anything else on Earth, you have to start not with what it does, but with how it is built. The architecture of an octopus is, by every conventional biological standard, wrong. Or perhaps more precisely, it operates by rules that biology itself had not fully written down until recently. An octopus possesses approximately 500 million neurons. That number alone is remarkable for an invertebrate. But the distribution of those neurons is what makes the arrangement truly strange. Only about 160 million of them reside in the central brain. The remaining 2/3, roughly 330 million neurons, are distributed across the 8 arms. Each arm carries its own dense, segmented nerve cord. Each arm, in a very real neurological sense, thinks for itself. When a researcher at the University of Chicago placed food in front of a restrained octopus, the arms did not wait for the brain's instruction. They reached, explored, gripped, and manipulated, processing sensory information, making motor decisions, and adjusting grip strength entirely at the local level. The central brain issued something closer to a general intention. The arms handled the execution. This is not a metaphor for efficiency. It is a literal restructuring of what a nervous system is allowed to be. A severed octopus arm, separated from the body entirely, continues to respond to stimuli. It recoils from noxious chemicals. It moves. It behaves with something that resembles purpose. The arm does not simply twitch, it responds. The neurons embedded along its length continue to function, process, and react long after the connection to any central processing has been severed. This distributed intelligence reshapes the question of where, exactly, the mind of an octopus actually lives. In humans, and in most vertebrates, the answer is obvious. In the octopus, it is genuinely unclear. The animal may not experience the world from a single, unified perspective the way a mammal does. Its awareness, if that word can even be applied, may be a chorus, not a solo. And then there is the genome. When scientists completed the first full sequencing of the octopus genome in 2015, what they found was quietly shocking. The genome contains approximately 2.7 billion base pairs, close to the scale of the human genome, and encodes an estimated 33,000 protein-coding genes, a number that exceeds our own. More striking still were the gene families that proved entirely novel, found nowhere else in the animal kingdom. Among these were dramatically expanded protocadherin gene clusters. These genes govern how neurons identify themselves, connect to one another, and wire into complex networks. In vertebrates, the expansion of protocadherins is closely associated with the development of intricate brains. The octopus arrived at the same solution through an entirely separate evolutionary lineage across hundreds of millions of years of divergence. The camouflage of the octopus is perhaps its most visible impossibility. Within milliseconds, the skin transitions from the muted tan of sandy seafloor to the mottled black and white of rocky substrate, and then, with equal speed, to the deep reddish brown of a patch of algae-covered stone. The match is not approximate, it is exact. The texture of the skin shifts simultaneously, papillae rising and flattening under muscular control to replicate the three-dimensional surface of the surrounding environment. Beneath the skin, three distinct cellular layers operate in concert. Chromatophores, elastic sacs filled with pigment, expand and contract under direct neural control, changing color across the visible spectrum within fractions of a second. Below them, iridophores reflect and scatter light, producing iridescent and metallic effects that no pigment alone could achieve. Beneath those, leucophores scatter all incoming light uniformly, generating the clean whites and pale tones that form the base of complex patterns. The brain, in effect, paints the entire skin surface simultaneously, in real time, through a neural broadcast that reaches tens of millions of pigment cells at once. But here is the problem that has occupied biologists for decades. The octopus has a single type of photoreceptor in its eye. 1. In human eyes, 3 types of cone cells, each tuned to different wavelengths, allow the brain to compare signals and construct the experience of color. The octopus, with only one receptor type, should be entirely colorblind. It should see the world in gradients of light and dark, unable to distinguish the red of a crab from the green of a sea anemone. And yet the camouflage it produces is, by any measure, color accurate. The leading scientific hypothesis at present involves the unusual shape of the octopus pupil, a narrow W-shaped aperture unlike the circular or slit pupils found in most animals. A 2016 study from the University of California, Berkeley proposed that this off-axis pupil shape allows different wavelengths of light to focus at slightly different depths on the retina, a phenomenon called chromatic aberration, effectively encoding color information within a single receptor type through the physics of focus rather than the biology of multiple receptor classes. The eye may be using the optical imperfection that most eye designs evolved specifically to minimize, and then there is the skin itself. Light-sensitive proteins called opsins, the molecular machinery of vision, have been found expressed in the skin of octopuses. Whether the skin is genuinely capable of detecting light and contributing to camouflage decisions, or whether these opsins serve some other function, remains an open and fiercely debated question. What is not debated is that the octopus solves the problem of color perception in ways that no other animal on Earth has been documented to use. The strangeness deepens when the environment changes. Most animals adapt to cold over generations through the slow accumulation of genetic mutations that alter protein structure across populations over thousands of years. The octopus does something more immediate and far more unsettling. When water temperatures drop, certain species of octopus, most dramatically studied in the California two-spot octopus and in Antarctic species, begin editing their own RNA. RNA, in the standard model of molecular biology, is the messenger. DNA contains the instructions. RNA carries a copy of those instructions to the cellular machinery that builds proteins. It is not supposed to be altered en route. It is supposed to be a faithful transcript. The octopus rewrites the transcript. Through a process of site-specific RNA editing, The octopus alters the functional properties of proteins in its nervous system without changing the underlying DNA. In cold conditions, the potassium ion channels in neural cells, critical structures that control the electrical signaling of neurons, are recoded at the RNA level, shifting their operating parameters to maintain proper neural function at temperatures that would otherwise slow or disrupt them. When researchers compared the RNA editing rates of Antarctic octopuses living in near-freezing water with those of tropical species, they found that cold, adapted octopuses showed dramatically higher rates of editing at precise locations known to affect channel behavior. This is not genetic adaptation in any traditional sense. It is something closer to real-time biological reprogramming. The animal does not wait for evolution. It reaches into the instructions for its own nervous system and rewrites them within its own lifetime in response to the temperature of the water. What this means for how we understand the boundary between an organism's fixed genetic identity and its capacity for self-modification is a question that biology is still working through. At some point in the night, the octopus sleeps. This, too, is not straightforward. A 2023 study published in the journal Nature documented what scientists had long suspected but struggled to confirm. Octopuses cycle through two distinct sleep states: a quiet phase characterized by stillness, pale coloration, and lowered responsiveness, and an active phase during which the skin begins to move. Pulses of color ripple across the body, patterns flash and shift, the arms twitch. The eyes, though sealed beneath the fused lids common to octopuses, show rapid movement beneath the skin. The active sleep phase of the octopus bears a structural resemblance to REM sleep in mammals, the stage in which the human brain shows its most intense and complex activity, and during which dreaming reliably occurs. Whether the octopus experiences anything during this active phase is a question science cannot presently answer. The neural correlates of dreaming in humans are relatively well mapped. In the octopus, whose brain organization differs fundamentally from any vertebrate, the same measurements produce results that are difficult to interpret. What the skin displays during active sleep may be residual motor output from internally generated neural activity, or it may be something more. The science does not yet know which. What makes this doubly striking is the context of the life surrounding it. An octopus lives, depending on the species, between 1 and 3 years. It does not grow old in the way most long-lived animals do, accumulating experience and relationship and territory over decades. It arrives fully formed in terms of its behavioral complexity, solves problems, navigates mazes, recognizes individual human faces, uses tools. Veined octopuses in Indonesia have been observed collecting coconut shell halves, carrying them across the seafloor and assembling them into portable shelters, and then, having reproduced once, it stops. The mechanism of this ending is not passive. After mating, males begin a rapid physical deterioration and die within weeks. Females lay their eggs, seal the den, and stop eating entirely. They guard the clutch for months without consuming food, circulating water over the eggs, cleaning them, tending them with continuous attention. The body dismantles itself. The optic gland, a structure analogous in some functions to the vertebrate pituitary, drives the cascade. When researchers experimentally removed the optic gland from female octopuses that had just laid eggs, the animals resumed eating. They lived longer. They left their dens. The gland was the signal, and the signal said, "This is where the life ends." The organism complied. Evolution produced this not as cruelty but as strategy. the resources invested in extended survival after reproduction offer, in the ecological niche the octopus occupies, a diminishing return. The young receive nothing from their parents' continued survival. The parent's body, already depleted by months of fasting and the physiological cost of egg production, represents a nutritional resource that the decomposing carcass makes available to the local ecosystem, and in some cases, potentially to the hatching young themselves. The death is not a failure. It is, in the coldest biological terms, the final act of investment. The evolutionary lineage behind all of this stretches back further than almost any other complex animal. The cephalopod ancestors of the modern octopus appear in the fossil record as far back as the late Cambrian period, more than 500 million years ago, before the first fish developed jaws, before the first vertebrate crawled onto land, before the continents assumed their current positions. Early cephalopods were shelled animals, their body plan not entirely unlike that of the living nautilus, which has survived largely unchanged for over 400 million years. At some point, roughly 300 million years ago, the lineage that would eventually produce the modern octopus made a radical, evolutionary commitment. It abandoned the shell. In a single long-term trajectory, the external armoring that had protected cephalopods for 200 million years was reduced, internalized, and in the octopus, lost entirely. The animal became soft, exposed, and vulnerable to forces it had previously been protected from. What it gained in exchange was flexibility, both physical and behavioral. A body without a rigid casing can compress itself through openings smaller than any of its rigid structures. An octopus the size of a human fist can pass through a hole no wider than its eye. The loss of the shell appears to have redirected evolutionary pressure toward intelligence, speed, camouflage, and behavioral complexity. A 328 million-year-old vampiropod fossil, described in research from the American Museum of Natural History, suggests that the ancestors of octopuses once possessed 10 arms rather than 8. The transition from 10 to 8 arms is part of a larger anatomical reorganization that accompanied the loss of the shell. The body plan was, in a very real sense, being rebuilt from the outside in. The octopus genome, with its anomalous gene families and its expanded neural wiring instructions, is the molecular record of that reconstruction. It shows a form of intelligence that did not emerge from the same evolutionary lineage as vertebrate intelligence, did not use the same developmental mechanisms, and did not build itself around the same neural architecture. Two fundamentally different biological experiments arrived at something that, from the outside, looks similar. Complex behavior, problem-solving, apparent awareness, and, possibly, something that happens during sleep that we do not yet have the language to describe. There are things science can measure about the octopus: the distribution of its neurons, the editing rate of its RNA, the layers of its skin, the structure of its genome, the pattern of its sleep. These measurements are real, precise, and remarkable. But there are other questions that measurement alone cannot reach. What is it like to think with 8 semi-independent minds? What does a colorblind animal perceive when it looks at the ocean and chooses flawlessly which colors to become? What passes through the nervous system of a female octopus in the final weeks of her life, when her body is failing and her eggs are still unhatched and she continues, without food, to tend them? We know the mechanisms. We are still learning what the mechanisms mean. And in the gap between those two things, between the biological fact and its full significance, the octopus remains, as it has for 500 million years, something that our frameworks do not entirely contain. The animal is real, the science is real, the strangeness is real, and some of it, for now, remains beautifully, rigorously unexplained.